Hi, everyone, and welcome to Microbial Minutes. This is American Society for Microbiology's every couple of week update on what's hot in the microbial sciences. I'm Julie Wolfe, the Scientific Communications Specialist here at ASM, and today is August 7th, 2018. It's been a couple weeks since our last session, but there's been a number of different stories making waves in different parts of the microbiology world. So what are we going to talk about today? We will start off with some drug-resistant infections from leeches, and then we'll move into a new study on the efficacy of alcohol-based sanitizer, which you may be familiar with carrying around in your purse or pocket. We'll then look at what foods cause the greatest amounts of foodborne disease from a retrospective study. And then there's a whole bunch of different Ebola updates. There's been a lot going on in the world of Ebola with outbreaks, as well as some studies about transmission and a new Ebola strain species. Excuse me. And then finally, we're going to end with a story about how viruses can counter the CRISPR attack. So let's dive right in. Our first story is from ASM's MBio journal, the premier journal at ASM, titled Low-Level Antimicrobials in the Medicinal Leech, Select for Resistant Pathogens that Spread to Patients. The take-home message here is that drug-resistant infections from leeches highlight the One Health concept. And the One Health concept is that all different ecosystems, whether we talk about that of the built environment, of the human microbiome, or of veterinary and or environmental microbiology, all of this is interconnected. And this story really emphasizes the interconnectedness of these different ecosystems. So a little bit of background information. It may be news to you. It was to me that leeches are used in reconstructive surgery. This has been a practice that has been on the upswing since the 1980s. Of course, we all recognize the medieval practice of bloodletting, but there are scientific studies that have shown that leeches can help after reconstructive surgery to keep the blood flowing so that there's an accumulation of too much blood, especially at poor sites of poor circulation. However, it was quickly realized after people started using these leeches that about a third of patients who are treated this way come down with infection. And the infection is normally from the leeches themselves. The leeches have a fairly simple digestive tract microbiome, but they do contain Aeromonas species, and Aeromonas is a potential human pathogen. So to counteract this or to prevent those infections from occurring, most patients who are receiving leech therapy are treated prophylactically at the same time with different antibiotics, including fluoroquinolone and ciprofloxacin. These are broad-spectrum antibiotics that should prevent any of those Aeromonas bugs that are in that leech gut from causing infections in an already vulnerable patient. But let's think about what happens when we take systemic antibiotics, right? So you take systemic antibiotics. These patients have to take them orally so that it enters the bloodstream and then reaches those areas where the leeches are feeding on the patient. But not all of those antibiotics are going to be used to kill a microbe. Most of those will be broken down by the liver before they're being excreted, but some of the antibiotics themselves will end up in the waste products of the patient. So it's going to be in the urine and in the feces. And particularly in places where there are a lot of patients who are taking prophylactic antibiotics, you can find trace amounts of these antibiotics in the wastewater, and some of those can even leach into the natural environment and natural waterways, both marine and freshwater waterways. And so this is where our story begins. There were 7 patients in the US, Canada, and France who experienced Aeromonas infections, and these Aeromonas infections were already resistant to the drug ciprofloxacin, which is normally given to the patients beforehand, right? What the question was whether these drug-resistant Aeromonas infections may have come from the leeches that were being used to help the patients recover from their reconstructive surgery. So to address this question, the researchers did several different experiments. First, they looked at how much ciprofloxacin it took in order to inhibit the growth of different Aeromonas populations. This is the minimal inhibitory concentration, or MIC, and they looked at this from both leech controls, so the leeches that did not contain any resistant Aeromonas species in their gut. They also looked at the 2012 to 2015 leech supply from a particular supplier, and they found that there was a similar MIC in the Aeromonas species carried within these leeches, as was seen in the Aeromonas, the drug-resistant Aeromonas in the patient wounds. So you can see that these are both fairly high, particularly when compared with those that were not drug-resistant, right? Now, in a court of law, we would consider this to be circumstantial evidence, merely coincidence, potentially, that they have similar MICs. And so the researchers undertook some deep sequencing assays across these different shipments, and they found that the vast majority of these resistant isolates were from the same genetic mutation. They had a substituted amino acid at the 83rd amino acid residue, which is normally a serine, and it was an isoleucine in the GIRE-A gene within the Aeromonas strains. And that was what conferred the resistance. And so this really helped to support the idea that the leeches were most likely the source of that drug-resistant infection in the patients. But the question still remained, where did those drug-resistant leech, the drug-resistant bacteria in the leeches come from? Because most mutations are selected for in the presence of drug, but outside of the presence of drug, there's not really a fitness advantage. And in fact, most of the wild type or non-resistant bacteria are likely to outcompete and grow faster and better than mutant strains. And in fact, the researchers then did some competition assays looking at how Aeromonas, either the mutant drug-resistant strain or wild type drug-sensitive strain, could colonize the gastrointestinal tract of these different leeches. And so they did some competition assays, and that's what I'm showing you here on the right-hand side. So competitive index indicates whether one of these different strains would be able to outcompete or grow better than another. They were ingested, or the leech ingested them at a ratio of 1:1, right? So for every mutant strain or mutant cell, there was a wild type cell. And should they be equally fit and able to grow at the same rates, they should remain at this line right here. But should the wild type be able to grow better, then the mutant would not be as competitive, and we would consider that competitive index in this case would be negative. And you can see without any drug, the wild type does not grow very well inside of the leech. It doesn't colonize the leech as effectively as the wild type does. With increasing amounts of ciprofloxacin, still at very, very low levels, these are comparable to what you would see in the environment. You can see that there are maybe a slight upward trend in having a few more of those mutant strains colonize the leech intestinal tract. But once we reach 0.01 micrograms per milliliter, there is a stark advantage to the mutant resistant bacteria. And so this supports the idea that perhaps the environmental trace amounts of antibiotics that the leeches may have been exposed to selected for a resistant strain of Aeromonas that then infected the patients when the patients were given the leeches as therapy, despite being given ciprofloxacin. So as I mentioned, this was a good measure or a good highlight of the interconnectedness of ecosystems, because these leeches, which are in natural waterways, can be exposed to some of the antibiotics that are in the wastewater and that are excreted from patients that are taking them systemically. This was written up in a number of places, including STAT, the magazine that covers a lot of biomedical and biotechnical research. And they highlighted very nicely, the author had this wonderful sentence that I thought really highlighted some of the importance of this particular study, stating that the work points to the butterfly effect of globalized medicine, with ideas and diseases ricocheting wildly through space and time, appearing, disappearing, and reappearing. And this was really a long-term case study, because these patients were throughout the world, right? They were in Canada, US, in France, at disparate healthcare centers, and being treated, like by different types of surgeons, and it really tying these cases together from a common shipment and common supplier of leeches took a while, because of course the main concern of any doctor or healthcare worker who's taking care of a patient is to make sure that patient recovers and then to find what the source of that infection is. There was also a quote from Ramanan Laxminarayan, the director of the Washington-based Center for Disease Dynamics, Economics and Policy, saying that this is 1/1,000,000th of the answer when thinking about how antibiotics released into the environment can end up affecting us, and that there are millions of other pathways that may lead to some of these coming back to bite us, right? So this is just one story that shows that all the different antibiotics that we're using and that are being released into the environment may have effects that we can't even predict at this moment. And this was highlighted additionally on Twitter through science news organizations from researchers involved in the study itself, as well as those who had been affected by leech related infections. And so that people really found this story to resonate with them in different ways. Moving on, we're going to next talk about a paper that was very recently released in Science Translational Medicine. This is titled Increasing Tolerance of Hospital Enterococcus faecium to Hand Wash Alcohols. Take-home message is alcohol disinfectant tolerance has been found in bacteria. And not just any bacteria. Enterococcus faecium is a good source of drug-resistant infections already. Enterococcus, this genus, is very good at picking up pieces of DNA and sharing pieces of DNA with other organisms, and that makes it a potential hazard for sharing things like drug resistance determinants. And so in this case, it's important to know that these alcohol-based disinfectants are particularly important in healthcare settings, both for hand rubs, right? So many of us carry those alcohol disinfectants that we'll use to put on our hands and rub on our hands if we aren't able to wash our hands before eating, for example. And this has been shown to be very effective as a good way to get rid of any potential pathogens. But this is particularly important for healthcare workers who might not have the time it takes to properly wash with soap and water. To properly wash with soap and water, one is supposed to lather for at least 30 seconds. That's singing happy birthday twice or thereabouts. But of course, if you have many different patients to see, you might not have time to sit and lather your hands completely and then rinse and dry them. And so a lot of stations in healthcare facilities will have these types of alcohol-based disinfectants that healthcare workers can quickly put onto their hands and rub as they're walking. And the use of these has really increased since the early 2000s, when this was found to be a suitable alternative. Now, the thought is that these alcohol-based disinfectants could not lead to resistance because of the mechanism of killing. The alcohol interacts with the lipid membrane of the bacterial cell. And so in theory, there should not be resistance found to these types of disinfectants. But what about tolerance? And that's where this particular study begins to look into this. So they looked, this scientific team looked at Enterococcus faecium isolates taken at different times. So you can see in this first figure I'm highlighting here on the right-hand side that 1997 to 2003, when those alcohol disinfectants were just starting to be used, what they did was to take different isolates that had been archived, and they grew them up again, and then exposed these isolates to approximately 70% isopropanol, which is the working formulation for a lot of these alcohols, these hand wash alcohols or the alcohol disinfectants. And they saw about a 2-log reduction. So that's about a 100-fold reduction in the number of colony-forming units, or CFUs. If they looked a few years later, 2004, 2009, and did the same thing, they similarly saw about a 2-log reduction in those isolates when they were treated with those alcohol disinfectants. But if they took more recent isolates from the years 2010 through 2015, they saw that the reduction in CFUs was halved, and that many of the different isolates were able to survive and were not killed by exposure to this alcohol. They also looked at this in a more realistic sense. So a lot of alcohols are used to disinfect surfaces. And so they took E-faecium contaminated mouse cages, and they washed them either with water, which was not expected to affect the Enterococcus that was in there, or with alcohol. And they did this with several different isolates. Then they would put mice into those cages and look at the number of mice that would somehow be exposed to the E. faecium and would become colonized in their gastrointestinal tract, right? And so if we look at this lower panel labeled C here, you can see that none of those different isolates, which are labeled down here, were affected by being exposed to water, right? We don't expect water to decrease the amount of colonization. All of the mice were colonized because there was no elimination of any of those bacterial cells. And if we look at earlier time isolates, such as this one in the first two bars and these with the green triangles here, we see that colonization after the cages were treated with alcohol is decreased somewhere between 70 and 40%. So not all of the mice become colonized with the E. faecium, presumably because there are fewer cells around that the mice are exposed to. But if we look at later isolates from this last time period, we see that decrease from using alcohol is not present, right? In fact, if we look at this isolate EF aus 0233, water and alcohol treatment equals the same amount of colonization by the mice into their gastrointestinal tract. And similarly, there is very little effect from that alcohol treatment in this later time isolate as well. And so this suggests that the E. faecium is not being killed as efficiently as it had been previously. And this made quite a stir on social media. It was shared by a group. This Battle Superbugs keeps really close tabs on drug-resistant organisms, and in this case, this is perhaps a tolerant organism. And it was written up in NPR's Goats and Soda blog, as well as The Guardian, which is a London-based news organization. And this was in The Guardian. The author, one of the co-authors, Timothy Steiner, was quoted as saying, "This is a wake-up call to infection control hospital teams around the world, that if you want to control the emergence of VRE, which is vancomycin-resistant enterococci, you need to do more than just rely on your alcohol-based disinfectants." And so this is something that I think a lot of people in infection control and in that clinical world are going to be paying attention to. However, this piece was highly criticized by hand washing expert Professor Didier Petit, who has undertaken many different campaigns to get healthcare workers to use both soap and water, but also alcohol-based disinfectants when they don't have time. And he was very critical not only of the Guardian piece, but of the science itself. And others within this field had really long conversations, including also the author of the Guardian piece. If you want to find it, it's not hard to find, as we're all open, transparent conversations on Twitter, with the consensus being that this is something that there's evidence within this paper that certainly suggests that enterococci can develop tolerance. What the mechanism of how they develop that has yet to be determined, but it's something that people should, in this world, should definitely pay attention to. And we will pay attention to it here on Microbial Minutes, should more be published on this. Moving on, we're going to next talk about a study from the Morbidity and Mortality Weekly Report, MMWR, from the CDC, titled Surveillance for Foodborne Disease Outbreaks in the United States from 2009 to 2015. Take-home message from this study is that retrospective study uncovers foodborne disease sources. And there are a number of really interesting tidbits to highlight in this particular piece. First, we should all remember that foodborne illness causes a lot of different illnesses annually in the US and worldwide. It's estimated to be about 9.4 million cases of illness. This is a very gross estimate, gross meaning rough in this case. And that's because lots of people who experience some sort of gastrointestinal upset from something that they've eaten don't go to the doctor. Very few of these illnesses result in hospitalization or death, the most serious sequelae of being infected, but it can result in people staying home from work and maybe taking a day or two to recover. Very few people even end up going to the doctor's office unless things get very severe. To get an understanding of where some of these foodborne illnesses are most likely to occur, the researchers in this case looked at outbreaks. So things that had been reported not only to healthcare workers, like people going to the doctor, but to authorities who track foodborne illnesses. And this was used to provide insight into the foods and the pathogens that are most likely to cause these types of disease. And so in some, they looked at 5,760 outbreaks, about 100,000 illnesses, about 5,700 hospitalizations, and 145 deaths that occurred due to outbreaks over this 6-year period. So if we look at the pathogen side of things, what they found is that norovirus is the most common etiology, the most often the source of an outbreak, with 1,130 outbreaks, or 38% of the outbreaks that had been caused by norovirus, and 27,000 illnesses. Salmonella came in a close second, however, with 896 outbreaks and 23,000 illnesses. So fewer outbreaks, but almost as many illnesses. But when we look at the severity of these illnesses, Listeria, Salmonella, and Shiga toxin expressing E. coli were responsible for 82% of hospitalizations and 82% of reported deaths, making these 3 bacterial causes the most serious of all that had been studied. When the researchers looked at where people were most likely to contract a foodborne illness, they looked at a number of different types of locations, and I'm just showing part of this graph or of this table from the report. Here you can see the locations that are bolded. Those are the ones that are the sum total of 100%, and then these are subtotals. So if we look here at restaurant, 61% of the outbreaks occurred due to a restaurant, while 14% due to catering or banquet facility. Only 12% of outbreaks were related from private home ingested foods. And this is in part because we as individuals take much more care. We have much more control over the food that we eat ourselves, right? So we know whether or not we've washed our hands after appropriate steps, such as after handling chicken, or thoroughly washed our produce to eliminate any potential contaminants there. And I think this is a good take-home message, that the food you eat at home, despite warnings of outbreaks in things such as cereal or various contaminated ice creams and things like that, you're much more likely to become ill by going to a restaurant or eating food that somebody else has prepared than food in your own home. Additionally, outside of looking at the pathogens and where people were likely to get sick, looked at what types of foods made people sick. So if you look at this as, say, like some sort of high school yearbook, the most likely to cause an outbreak were fish, dairy, and chicken, which I think many listeners probably would have guessed. But if you look at those which are most likely to cause a severe disease, and in this case, hospitalization, chicken moved from 3rd to 1st. And this is something that was picked up by a lot of outlets, as we'll discuss. Chicken, pork, and seeded vegetables, things like sprouts, which again, many listeners probably know that sprouts are really hard to wash and eliminate all of the potential contaminants. And so those were number 3 with most hospitalizations. Multi-state outbreaks, where more than one state was involved, comprised only 3% of the outbreaks that were studied, but accounted for 11% of illnesses, 34% of hospitalizations, and 54% of deaths, in part because these multi-state outbreaks are a lot harder to source because perhaps one source of chicken or produce may be distributing to many different restaurants or grocery stores or other locations, and can be harder to pinpoint and stop those multi-state outbreaks. All right, as I said, this was picked up by a number of different outlets, so we have a question. Thank you, JA. Where does Staphylococcus aureus rank in the foodborne illnesses? I don't remember, but we are linking to all of these different reports, and I actually recommend that everybody read this one. This was one of the most readable reports because they do a beautiful job of a lot of tables, such as this, talking about where people get sick, but also some really beautiful data visualization showing what states were most likely to have foodborne outbreaks, what years. And so I encourage listeners to find that link, which we'll have down below, and go have a look at that. So a couple of resources. This was picked up by a number of different outlets because everybody likes to talk about foodborne disease. We all eat food and we want to stay healthy. So this was picked up by Self Magazine. It was picked up by the Today Show. In the Today Show's write-up of their video, video clip, they highlighted that the CDC report shows the government inspectors and industry need to do more to protect consumers from unsafe chicken. You can see chicken was really the highlight of a lot of these stories, even though a number of these different foods can cause foodborne illness. So don't think that only chicken. Chicken was in the lead, but not by a lot, right? And so this is shared by folks who are in the industry and are interested in this. This was shared by Maren McKenna, who has just written a book about chicken and its role in spreading antibiotic-resistant infections. And I want to point you to a couple of ASM resources, an interview with Eric Brown, who uses microbial genomics at the FDA to pinpoint the sources of some of these food outbreaks. We'll link below. And subscribe to the ASM channel for a food parasites interview, which is coming out in just the next couple of days, to learn more about the potential food parasites that can contaminate our foods and how to avoid them. All right, we're going to move on to the next study, and this is a collection of different studies. So this is our Ebola outbreak section, where I want to highlight a new virus species, some updates about a second outbreak that's ongoing in the Democratic Republic of Congo, and persistent transmission of Ebola a year after recovery. So we're just going to spend a quick slide on each of these 3 points, but I think that there's been so much going on in the world of Ebola that we needed to spend some time on all of these different aspects. So let's first talk about the new Ebola virus that's been found in Sierra Leone. This was announced in a very non-conventional way. EcoHealth Alliance is a nonprofit that's based in Hudson Valley, New York, and they linked to a story that was in Punch, which is the website of an African newspaper, where this news release was first published. And the news was that Bombali Ebola virus looks like a new Ebola species, not just a strain, but a new species. Whether this Ebola species can be transmitted to humans is yet unknown. There have not been cases of humans contracting this disease that we know of yet. However, this particular virus can infect human cells in vitro, suggesting that it may be something that can infect humans, and whether it infects humans and causes disease or not is, of course, unknown as well, as there are Ebola species that can infect people subclinically. This species, the Bombali Ebola virus, was found in an Angolan free-tail rat of the Mops genus in Sierra Leone. It was named Bombali after the district in Sierra Leone in which it was found. And it was not found as a live virus in this bat, but enough viral RNA fragments were sequenced that allowed the researchers to tie together the genome. And this was announced in a non-conventional way, as I said, because the government of Sierra Leone wanted to ward off rumors about this particular virus and start public health campaigns. For example, do not eat bats because they may contain this particular virus. There's a paper that will have a little more scientific data about this species that is currently under embargo and should be published shortly. But should that peer-reviewed paper hold up, this Bombali Ebola virus will join 5 known Ebola species, including Zaire, Bundibugyo, Sudan, Forest, and Reston. Those first three with the star have been known to cause outbreaks in humans. And that last one, Reston Ebola virus, is the species that's discussed in Outbreak. No, I'm sorry, the Hot Zone, and I guess was turned into the movie Outbreak, and is the Ebola species that does not cause disease in humans overtly. All right, so we will look out for when that actual scientific paper is published, but this just hit the news waves in the last couple of days, so I thought it was something that you guys would be interested in. Moving on, you may have heard in the news that there's a number of outbreaks that have occurred even within the last couple of weeks. So our last session was July 23rd. On July 25th, the World Health Organization announced that the Ebola outbreak that had been ongoing and previously in the summer was officially over. And that's because 42 days, which is 2 incubation periods of 21 days, had passed since the last possible exposure to a confirmed Ebola virus disease case. So that was good news and something that I planned on sharing. However, unfortunately, a few days later, a new outbreak was detected. And just in the last couple of days, the World Health Organization has released some stats about the ongoing disease, which appears unrelated. So it's not the same virus that was being passed in the earlier summer DRC outbreak, but it is still in DRC, Democratic Republic of the Congo. The good news is that this virus, although it has already passed into a number of individuals, so there are 43 confirmed and probable cases, according to Helen Branswell, who summarized some of the stats from the World Health Organization. 13 lab confirmed cases, but there have been 33 deaths, but 30 of the dead are people who died and were buried before the outbreak was recognized. So this virus may have been passing from person to person through human body fluids, which is how it's passed it between people. It may have been circulating before it was recognized as Ebola, since, of course, some of the symptoms of Ebola can be a little hard to distinguish from other types of hemorrhagic fever diseases. The cases are illustrated here in the map of the DRC. It's in this north eastern part of the country. And as I mentioned, there is a little bit of good news, and that's that the species and strain that are circulating are the same as the vaccine strain, which we have discussed in a previous Microbial Minutes session. And so I think that the healthcare workers who are going to attack this outbreak and do their best to stop it quickly will hope to do some of the ring vaccination, which will hopefully stop the spread of the disease quickly. And we will bring updates in the next couple of sessions on how that progresses. Finally, I did want to talk about the Lancet Infectious Diseases paper that was published a few weeks ago, titled Persistence of Ebola Virus After the End of Widespread Transmission in Liberia, an Outbreak Report. So we all remember the 2014 to 2016 West African Ebola outbreaks, the largest outbreak to date, with tens of thousands of people who are affected. And let's keep in mind that Ebola virus is spread only through bodily fluids. So there's a lot of studies that are still ongoing from all the data that was collected. We've never had such a large population that's been infected that we can study what the sequelae are and what the transmission or some of these other factors or characteristics of the disease itself. We do know that survivors of Ebola virus can live for decades. After all, there were outbreaks in the 70s, and there are still survivors who are living healthily and still making antibodies against Ebola virus, as we also talked about in the previous session. And we also know that virus persistence can occur. We learned this in this large outbreak when healthcare workers who recovered had unusual things happen, such as one healthcare worker whose eye changed from blue to green and was found to be harboring Ebola virus inside of the eye fluid. There was sexual transmission that was discovered in the last outbreak, where a healthcare worker went home after recovering and passed Ebola onto his wife by having unprotected sex, and she contracted it that way. Although there has not been any record of female to male sexual transmission. That doesn't mean it can't occur, but there has not been observed yet. And the virus can persist in semen for quite a long time. One man has had 565 days, which is, really on the order of years. And so this story begins when a 15-year-old boy in Monrovia in 2015 tested positive for Ebola. And the question was, where did he get this illness? Where did he get this virus? Now, his father and 3 siblings were also sick around the same time in 2015, but they got sick a little bit after, and so were not thought to be the source of the illness. However, his mother had been sick with Ebola in 2014, but she had recovered, and in the meantime had even had another child. And so to get to the bottom of this, there were a number of different types of clinical pathologies that were tested. They looked at the serology to look at the antibodies. This paper really reads almost like a CSI investigation, trying to find how this transmission had occurred to this young boy. And it turned out that Ebola genome sequencing was able to best answer that. There's a summary of some of those connections between the members of the family and the mother, but the sequencing, which is shown over here on the very far right-hand side, is where we see the connection between the mother's Ebola genome sequence, because she was harboring yet Ebola, and her sons, and those are depicted down here. So there were others involved in this connected circle who had a related case, but there were still some differences. There were some other Liberian cases as depicted over here. Other Liberia, this is all the same country as where Monrovia is found, but you can see that the connection between individual ANC, which represent the mother-son duo, are very closely related. And so the most likely source for that 15-year-old boy to get sick was his mother. And this really highlights how much we have yet to learn about this disease, and hopefully some of these other case studies and ongoing research studies from the large data sets of all these people that were sickened will help to prevent things like this from occurring, these kinds of transmissions from occurring, should future outbreaks of this size occur. All right, we are going to move into our last story. And so we're going to talk about 2 back-to-back Cell papers that were published a couple weeks ago. First is titled Bacteriophage Cooperation Suppresses CRISPR-Cas3 and Cas9 Immunity. And the second is titled Anti-CRISPR Phages Cooperate to Overcome CRISPR-Cas Immunity. Take-home message, bacteriophage defend against CRISPR together. And really, I like this story because it shows that there is always one-upmanship in host pathogen interactions, where when the host thinks that it's found a way to defend itself, the pathogen has found a way to defend itself against the defense and continue infection of some sort. And so let's remind ourselves that bacteria have a type of virus that can infect them. I'm depicting a schematic over here on the right-hand side. Bacteriophage or phage inject their genetic material into the bacterial cell, which is depicted down here, and that genetic material often will incorporate into the bacterial genetic material, where the bacterial polymerases and ribosomes will make the viral genetic material and the viral proteins necessary for new viruses to be made, and then this will eventually make so many viruses that they will break open the cell and kill the bacterial cell. There's a lovely picture of some bacteria, many, not just some, but many bacteriophage, which are attached to this cell, which is just showing the top part of the cell here. Of course, it would be much larger than this. And to defend against bacteriophage, putting their genetic material into this bacteria and then eventually killing the cell, there is a defense system called CRISPR or CRISPR-Cas9, in which the bacteria, which have survived a phage attack, take pieces of the previous attacker's genomes and encode those into the DNA. And should that same virus sequence be recognized as the virus is injecting its genetic material, that viral genetic material will be recognized and cleaved. And so it won't be able to integrate and it won't be able to make new variants, and it's the end of the line for that particular bacteriophage. So these stories begin when the research teams both independently noted that multiplicity of infection could be used to predict success of phage infection. So multiplicity in infection means the number of bacteriophage particles to the number of potential bacteria host cells. If we look at this schematic, this is a pretty low MOI because there's only 2 viruses for one particular cell in this schematic. But if we look at the electron micrograph, we see this is a really high MOI, meaning that there are lots and lots. I mean, there's 20 or 30 phage in this particular section, and we imagine it goes all the way around and in all different 3 dimensions. So this high MOI was found to be more likely to produce progeny viruses successfully, and both research teams wanted to ask why. These being Cell papers, they have these lovely graphical abstracts, and we're going to use these to discuss the data. And so there were 2 different approaches which were used. The first team, the Borges et al., used a genetic approach. So some phages have a protein called an anti-CRISPR protein, which is thought to be able to inhibit this CRISPR-Cas defense system, but doesn't always work. And so what they did was to engineer a phage that expressed anti-CRISPR proteins, but can't replicate. It can't make any progeny viruses. But they would add this engineered phage with some wild-type phage that both had the anti-CRISPR proteins, but also could replicate. And then they could test what that MOI effect was. And what they found was that when the anti-CRISPR protein expressing phage was added with this wild-type phage, even a low MOI could produce progeny variants, right? So somehow this anti-CRISPR protein, having more of it, helped to make viruses, even if there weren't very many viruses around. The second team, Landsberger et al. Over here, used a modeling approach. And they found that they were only able to get their actual results to match the model when the bacteria in their model were hypothesized or posited to become immunosuppressed from an initial infection with phage expressing anti-CRISPR proteins. And so they theorized that these low density of anti-CRISPR phage did not produce enough of this initial anti-CRISPR protein in order to make those bacteria become immunosuppressed. And so putting these 2 models together in a very nice news and views from Nature site that we will also link to, as well as the original studies, what the model for this interaction is that the first phage will come along and inject its genetic material into the cell, and quickly some of those anti-CRISPR proteins will be expressed, and that will start to inhibit some of the CRISPR-Cas interaction, but not all of them. And so this virus is not going to successfully infect the cell. However, these anti-CRISPR proteins are going to stick around, as you see here, and that will allow a second phage of the same type to inject its genome. And while these anti-CRISPR proteins are making this particular cell immunosuppressed, this second phage, or a later phage, would be able to successfully replicate inside of that cell. And so this was written up mostly in technical outlets such as the Genetic Engineering and Biotechnology News, where the senior author of the first study here and the discoverer of those anti-CRISPR response proteins, Bondi Denomi here, said that what we think is happening is that the first phage acts like a kamikaze phage. It gets destroyed, as you can see in this depiction here, but along the way it starts to produce a few of these anti-CRISPR compounds that will neutralize some CRISPRs and therefore help its kin, the subsequent phage infection. And this was also released in a press release from the institution where Bondi Denomi works, and was also discussed quite a bit on social media. People who study this were talking about it on Twitter quite a bit. All right, so that's going to wrap it for today's Microbial Minutes sessions. And so what have we discussed in the microbial sciences? We talked about the drug-resistant infections from Leech, leeches used in surgery, which highlight the One Health concept of connectedness between ecosystems. We talked about alcohol disinfectant tolerance, which has been discovered in bacterium in E. faecium. We talked about the retrospective study, which has uncovered the sources of a number of foodborne diseases. And I see there's another question about this particular study, also from JA, saying that since Listeria likes cold, is it safe to assume that most dairy food poisonings is caused by Listeria? And I think that Listeria can be a major contaminant in dairy, but it's not the only one, because some of the contaminants also come from the udder of the cow and are not only Listeria, but you can get other types of bacteria that are naturally found in that udder that can contaminate those subsequent dairy products. We also talked about Ebola with a number of updates. We talked about a new virus species that was found. We talked about this second DRC outbreak that has only just started in the last couple of weeks and is continuing, and hopefully we'll have good news about that soon. And we talked about persistent transmission a year after recovery. And finally, we talked about bacteriophage and how they defend against the bacteria CRISPR system by working together. I'd like to thank you guys for listening. I'd like to thank Ray Ortega for helping to produce this session. And if there are any questions after this live session, feel free to leave a comment, and I will be happy to come and answer these questions later. All right, don't forget to subscribe, tell your friends, and we'll talk to you next time on our next Microbial Minute session.